Hey guys, Mr. P. In this video, we're going to talk about the absorption and action spectra of photosynthesis. So in the last video, we talked about the photosynthetic equation, looking specifically at the inputs and outputs. We looked at a balanced equation. We compared it to the cell respiration equation. We talked about redox reactions and how one of the reactants is oxidized and one is reduced, and what that oxidation and reduction results in on the product side. And now we're going to talk specifically about how pigments that are contained within the plant absorb light and how it promotes photosynthesis moving through the reaction. So in this video, we're going to highlight three objectives, C1, 3, 4, 5, and 6. C1, 3, 4 states separation and identification of photosynthetic pigments by chromatography. That is a lab technique that you should be familiar with. We will actually do that in class as well. C135 is absorption of specific wavelengths of light by photosynthetic pigments. And then C136 looks at the similarities and differences between the absorption and the action spectra when looking at a graph. Again, this is an application of skills which we will do in class as well. So, three objectives, absorption and action spectrum. Let's go. So when we focus on a plant at the organism level, you see leaves and stems and sometimes roots and shoots and flowers and fruits and all of the things that make up the organism of a plant. But when we dive into the actual cellular structure of a plant, you'll notice that when looking through a light microscope in this case, you see a bunch of individual cells that have some irregularities, but for the most part they're pretty consistent in a nice, neat, uniform tissue layer. If you zoom in to one of the cells, you'll notice that there are a variety of these little green circular structures. Each one of these is a chloroplast. Now, the chloroplast, if you remember from our cell unit, is an organelle that utilizes photosynthesis and incorporates pigments, which is why it appears green. So many of these chloroplasts packed into a little area makes the entire organism appear green. And all of those pigments are contained within this organelle called a chloroplast. Now, if you dive in or focus on one individual chloroplast, you'll see a variety of structures. All chloroplasts have two membranes. You have an outer membrane, which is the outside most membrane. You have an inner membrane, which is the membrane inside of the outer membrane. In between the outer and inner membrane, you would have this inner membrane space. Inside of the inner membrane is a fluid called the stroma. Inside of that fluid is a set of flattened discs called thylakoids. Each one of the individual discs would be a thylakoid. These thylakoids are flat to increase surface area and decrease volume. It's one of the adaptations that this particular cellular structure employs to make itself more efficient. All of these individual flattened discs called thylakoids are organized into stacks of those discs, and a stack is called a granum. And if you look at one of these thylakoids, or a cross-section of the thylakoid, if we were to zoom in to this thylakoid, there actually is a thylakoid membrane, and there is a space on the inside of one of those discs that is referred to as a lumen. We will focus on this particular structure, the thylakoid, when we get into a later video called the light-dependent reactions, because all of the light-dependent reactions occurs within this thylakoid membrane. But for now, this cellular structure is composed of a variety of these flattened discs called thylakoids stacked up in stacks called grana, and the sole purpose of those discs is to increase surface area, decrease volume, to make those discs a solar panel, essentially, which absorbs light to facilitate photosynthesis as long as light is present. Each one of these thylakoids contains a variety of pigments. The pigments that the plants contain would be chlorophyll A, chlorophyll B, carotenoids, and xanthophylls. All of these are pigments. All of them are going to be in various concentrations, and all of them play an important role in photosynthesis as a whole process. So, when we look at or start to look at these individual plant pigments, we employ a lab technique called plant pigment chromatography. Plant pigment chromatography requires us to take a strip of chromatography paper, put it into a graduated cylinder with a little bit of chromatography solvent at the bottom. We typically will put a dot of plant pigment, basically a little dot of plant juice that we extract from leaves of a particular plant here, making sure that it is actually outside of and not submerged in this chromatography solvent. As the paper strip is dipped into the chromatography solvent, the solvent will start to move up that paper strip by capillary action. It's one of the properties of water, which the solvent is acting like. And as that solvent moves up this tube, it's going to begin to pull these pigments with it. Now notice the pigments are starting to separate into individual bands, and they're going to separate based on molecular size and how quickly, or the rate at which they move by way of that capillary action at that movement of solvent. So when we look at the strip of paper, again, this little kind of ghosted image is where that plant pigment would have started. It would have originated here. And then as the juice, or as this plant pigment extract begins to move up with this solvent, it will start depositing the different pigments at a certain place on that chromatography paper based on the molecular size of that particular pigment. And we can then begin to calculate what is called RF values. Now, like I said on the last screen, chlorophyll B, chlorophyll A, xanthophylls, and carotenoids, or carotene, are the 4 pigments we're going to focus on for this particular class. Each one of them separates out. They are all individual pigments, and they are individually sized, meaning they differ in size, which is why they move different amounts. Obviously, chlorophyll A and chlorophyll B are going to be more similar in size than xanthophylls is to the chlorophylls, and definitely the carotenoids are to the chlorophylls. Carotenoids would be the smallest, xanthophylls would be the next biggest, chlorophyll A would be the next biggest, and chlorophyll B would be the largest of the pigments in this particular sample. So the paper is referred to as the stationary phase because it doesn't move throughout the entire chromatography process. We dip it into the solvent and then it remains completely stationary. It doesn't move. The only thing that is moving is the solvent as it moves up the chromatography paper. The mobile phase is the solvent that moves up that stationary phase, which is the paper, by capillary action and hydrogen bonding and cohesion and polarity and all of those things that we talked about when we did the properties of water. As the mobile phase moves through the stationary phase, it carries the components of the mixture being analyzed with it. Again, the components of the mixture are all of these pigments that all exist in the plant extract while it is one uniform solution, but as it moves, they do separate. During the movement, pure substances separate from each other because they have different migration rates across the stationary phase, and those migration rates, or RF values, are going to differ based on the molecular weight and the molecular size of the particular pigments. So, a paper chromatograph, which is the actual paper, the chromatograph is the product of the technique, which is chromatography. This paper chromatograph obtained from a leaf of a plant showing the various pigments that are necessary for photosynthesis. Again, the pigments would be the carotene or the carotenoids, the xanthophylls, the chlorophyll A, and the chlorophyll B. All 4 of those pigments are present in the leaf stain or in the leaf extract. Each of the pigments migrated at a different rate to produce the separate bands, which are labeled with the name of the pigment here. The RF value is the ratio of the distance traveled by the pigment, specifically, to the distance traveled by the solvent. You can look really carefully. The solvent basically stopped here. So if the leaf stain started here, we can measure with a ruler the total distance the solvent moved, and we can compare the total distance the pigment moved, and that helps us calculate the RF value. So this chlorophyll B, we would measure this distance, and we would put it over this distance, which is the solvent movement, and we would get this. Whereas if we measured this distance and compare it or put it over this distance, then you get this value. Chromatography allows us to determine the pigments present in chloroplast, again, because we can actually separate and isolate them. Pigments with higher RF values are more soluble in the solvent being used, and they are often smaller in size. Again, the smaller the molecular weight and smaller the molecular size, the larger the distance the pigment travels. And so if you looked at this pigment by molecular size, this pigment would be much smaller than these pigments, which stay very close to where they started. Larger pigments or larger molecules don't move as far as the smaller pigments do. Higher RF value pigments have a lower affinity for the paper in the chromatography activity, and so they don't get stuck where they are or struggle to move as much like the larger molecules do. Plants use the visible part of the entire electromagnetic spectrum. So when you think about the electromagnetic spectrum, we know that visible light, Roy G. Biv, red all the way to violet, is part of a much larger electromagnetic spectrum, much of which we don't actually see. We only see a very small wavelength band of the entire electromagnetic spectrum. Obviously, if you go to the right of red in this case, or the wavelengths get larger than red, we get into the infrared, we get into microwaves, which we obviously use to heat up and or cook food, and then you get into radio waves, which are the largest of the waves within this electromagnetic spectrum, and we use those to do long-range communication. If you go to the left of our violet and we start getting smaller on our wavelengths, you get past violet to ultraviolet. You get into X-rays, which we use to take pictures of bones and internal hard structures, and then getting into gamma rays, which we even use to see smaller structures and or soft tissue. But while we're talking about the electromagnetic spectrum, it's important to note that plants use the same part of the electromagnetic spectrum that our eyes see. So plants are going to specifically use this part of that electromagnetic spectrum, and plants will utilize everything from the red end of the spectrum through the green middle of the spectrum all the way to the violet or blue end of the spectrum. So plants will utilize this entire visible light spectrum to help facilitate photosynthesis in order to produce the glucose they need to survive. When we take that visible part of the spectrum and plot it on a graph and start to look at kind of two different variables, the absorbance and photosynthetic rate, we start to plot lines that peak in the violet and blue range, and they peak in the orange and red range. The amount of light absorbed plotted against the wavelength of light produces the absorption spectrum of that pigment. And so these kind of muted bands are going to be the absorption spectrum. Okay, this is absorption spectrum of chlorophyll A. This is absorption spectrum of chlorophyll B. This would be absorption spectrum of the carotenoids. And when we look at this absorption spectrum, this is the amount of light absorbed by those particular pigments. So this entire part of the electromagnetic spectrum would be the visible spectrum. And we know that the visible spectrum runs from red through orange, yellow, green, blue, indigo, and violet. That is the Roy G. Biv. We know from art class or science class that Roy G. Biv is the part of the electromagnetic spectrum which is visible light. And so if we focus on chlorophyll A, we can see that chlorophyll A is going to absorb well the violet and indigo portion. That's where it peaks, violet and indigo, and it also peaks on the orange, red, and red end of the spectrum. So chlorophyll A is going to absorb the two extremes, okay, the violet and the red. Chlorophyll B is going to absorb the indigo and the blue, and it's also going to absorb the yellow and the yellow-orange and the orange. So just inside the extremes. Carotenoids are going to absorb the blue and the blue-green and not absorb anything else on the right side of this electromagnetic spectrum. And so why does a plant have or need multiple pigments? Well, each pigment absorbs a particular frequency or wavelength size the best. So chlorophyll A has been adapted to absorb the extremes. Chlorophyll B has been adapted to absorb just inside the extremes. And carotenoids have been adapted to absorb the things that the chlorophyll A and chlorophyll B can't do well. And so if a plant contains multiple pigments, it can absorb more energy than it did with just one pigment. So the action spectrum indicates the rate of photosynthesis at different wavelengths of light. Now we're focusing on this bolded line. Notice that it follows the same trend as our absorption spectrum. Notice that where our absorption peaks, the action peaks, and where the absorption drops, the action drops. But they are not identical. Absorption indicates how much of that light frequency is absorbed by those pigments. The action spectrum is how efficient, or the rate, how quickly photosynthesis is happening within that plant. Two of the most common ways to calculate the rate of photosynthesis is measuring the oxygen production or carbon dioxide consumption over time. So if we put a plant into a container that was sealed and we used an oxygen probe or a carbon dioxide probe and we actually gave it different frequencies of light, it being the plant, we could actually look at the rate of oxygen being produced, or we can look at the consumption of carbon dioxide over time. And frequencies of light that cause photosynthesis to go up are going to have higher peaks on this action spectrum than frequencies of light, like in the green, that have lower rates of photosynthesis. At wavelengths where there is higher percentage of light absorption, the rate of photosynthesis is also high, and that makes sense. The more light energy a plant absorbs, the higher the rate of photosynthesis is overall. And when absorption of light by a pigment is low, like in the case of green, which is very low in terms of amount of light absorbed, the contribution of that pigment to the rate of photosynthesis will also be low. So if a plant absorbs the light well, it's going to do photosynthesis well, and when it doesn't absorb the light well, it's not going to do photosynthesis well. So what are the similarities and differences between the absorption and action spectrum? Again, the absorption spectrum would be these muted colors, and the action spectrum would be this bold line. The absorption spectrum varies depending on type of photosynthetic pigment present. Remember, each one of these lines is a different photosynthetic pigment. This was chlorophyll A, this is chlorophyll B, this is the carotenoids. Each one of them has a different absorption spectrum. But when comparing the absorption spectrum to the action spectrum in this case varies also depending on the type of photosynthetic pigment present. If these three pigments are present, the absorption spectrum and action spectrum are going to basically mirror each other. Where the absorption spectrum peaks, the action spectrum will peak, and when they both fall, they will both fall. Absorption spectrum is going to represent the amount of light energy being absorbed by the photosynthetic pigment. Again, that's why it's called the absorption spectrum. The action spectrum represents the rate of photosynthetic process being carried out by the plant. Again, this is the action. It's the action of photosynthesis, and it's the rate that that process is happening in the plant. Absorption spectrum would be for the plant. This spectrum represents light absorbed by all the pigments present. Action spectrum for the plant, this spectrum represents the rate of photosynthesis as a result of all pigments present. Again, all of these pigments constitute one action spectrum, which is why there's one line indicating the action spectrum. The absorption spectrum is chlorophylls A and B have a high absorption of light energy in the violet, blue, and red light wavelengths. The action spectrum, chlorophylls A and B create a relatively high efficiency rate of photosynthesis, especially in that same band of color, the violet, blue, and the red light wavelengths. Absorption, the pigments like the carotenoids absorb light energy at different wavelengths compared to chlorophylls A and B. So it is a complement to the normal pigments, chlorophyll A and B. And the action spectrum, the pigments like the carotenoids allow photosynthesis at different wavelengths that the chlorophylls A and B cannot absorb well at. It just helps make the plant more efficient using wavelengths that the chlorophylls A and B can't use. Other pigments are not as efficient at absorbing light energy as chlorophylls A and B, and that leads to other pigments not being as effective at achieving high rates of photosynthesis as chlorophylls A and B. Chlorophylls A and B are the primary players in photosynthesis, which is why the absorption and action spectrum peak basically over the chlorophyll A and B's peaks. So how do these pigments and light absorption work? So within a chloroplast, specifically within the thylakoid membrane of a chloroplast, there are all of these individual pigment molecules. Every single one of these little dots or circles is a pigment molecule. When light energy comes in, and these represent light energy, both the blue end of the visible light spectrum and the red end of the visible light spectrum, these pigment molecules act like mirrors, and this light energy is going to be kind of analogous to a laser. And so if you shined a laser on a series of mirrors, you can bend or orient the mirrors to cause the laser to bounce off of each other and focus on a specific point. The specific point in this entire array of pigment molecules is called a reaction center. When we get into our light-dependent reactions, we will start to learn that photosystem 1 and photosystem 2 are two bundles of pigment proteins, much like this is, and each one of these photosystems has its own reaction center. Photosystem 2 has a reaction center P680, which represents phytochrome 680, and the reaction center specifically within photosystem 1 would be phytochrome 700. So we have two reaction centers, P700 and P680. Each one of those is going to be a charging station that allows us to energize electrons. Again, those electrons are going to be taken from a normal energy state to a high energy state. They essentially charge the electrons. The plants are utilizing the sunlight energy, focusing that energy through a series of mirrors. Again, this is an analogy. Focusing all of that light energy on one reaction center, which energizes these electrons or takes them from a normal energy state to an energized or a higher energy state. And then these high energy electrons allow us to do something with that energy. They actually can cycle between high energy and low energy. And when they do that, they release energy, and it's this energy that actually is going to allow us to facilitate efficiently our light-dependent reactions, which helps us produce two compounds, ATP and NADPH, which will be transferred from the light-dependent reaction to the Calvin cycle, which will eventually allow us to make glucose. So we're going to get into the actual intricacies of the light-dependent reactions in a later video. I just wanted to put this in as a way of identifying what the purpose of the pigments and light absorption are. And so pigments are going to be contained within the thylakoids. They're going to absorb light, both red and blue. They're going to send the light through a series of pigment molecules, much like a laser bouncing off mirrors, focusing on that reaction center. That reaction center is going to energize normal electrons to a high energy state. Those high energy electrons are going to be utilized to do molecular work within the light-dependent reactions, which will produce both NADPH and ATP for the Calvin cycle. That's it for this particular video. If you learned something, give it a thumbs up. Subscribe to the channel. If you have a question, leave it in the comments. See ya.